Looking at this diagram, you can see the functional group of a carboxylic acid is built from two distinct parts: a carbonyl group, characterized by a carbon double bonded to an oxygen, and a hydroxyl group, where an oxygen bonds to hydrogen. This specific RCOOH structure branches out into a wide taxonomy. We have aliphatic acids, like the acetic acid found in vinegar, aromatic acids featuring benzene rings, like benzoic acid, and dicarboxylic acids containing multiple carboxyl groups. When you place a highly polar hydroxyl group directly adjacent to an electron-hungry carbonyl, the resulting structure is significantly more reactive than either group standing on its own. Carboxylic acids form hydrogen bond dimers, a dual connection between two molecules. Breaking these strong bonds requires massive energy, resulting in much higher boiling points than comparable alcohols. This bonding also dissolves lower molecular weight acids in water until the nonpolar chain grows too long. The macroscopic physical properties you can measure in a beaker, the high boiling point, and the specific limits on solubility, are direct, predictable consequences of how effectively these molecules lock together as dimers. Carboxylic acids willingly donate a proton, a hydrogen ion, in solution. Alcohols and phenols technically possess the same ionizable hydrogen, but they hold on to it. To understand why carboxylic acids act as stronger acids, we have to look at what happens to the molecule after the proton leaves. This diagram holds the answer. An alcohol's ethoxide ion traps the negative charge on a single oxygen, making it unstable. Phenols spread it unevenly. But look at the central acetate ion. Through resonance stabilization, the negative charge spreads perfectly across two identical oxygen atoms. We can amplify stability using the inductive effect by swapping hydrogen for highly electronegative chlorine atoms. These chlorines act as an electron vacuum, pulling density away from the oxygen and delocalizing the negative charge further. More electron-withdrawing groups create a stronger acid. The central rule of acidity here is strict. The strength of an organic acid is entirely dependent on how effectively the molecule can stabilize the negative charge after it loses its proton. In a laboratory setting, you won't be able to physically see resonance structures. Instead, we use chemical fingerprinting, exploiting these specific theoretical vulnerabilities through physical lab tests to identify invisible molecules. Our first diagnostic is the sodium bicarbonate test. Because carboxylic acids are strong, they decompose weak bases like sodium bicarbonate into water and carbon dioxide. If you add the base and see rapid gas effervescence, you have successfully confirmed the presence of a carboxylic acid. Our second diagnostic uses ferric chloride. The reaction requires a strictly neutral medium. You convert the acid to a salt with ammonia, then boil it. Once neutralized, it acts as a color identifier. Benzoic acid yields a buff precipitate. Salicylic acid forms a violet complex. These two laboratory tests operate as a sequence filter. The bicarbonate effervescence proves you are dealing with an acid. The ferric chloride colors determine exactly which acid it is. Now consider an aliphatic compound like formic acid. It doesn't yield a distinct color with ferric chloride. Pause and ask yourself, what unique structural feature separates formic acid from acetic acid? Formic acid has a hydrogen atom directly attached to the carbonyl group, making it look and behave structurally like an aldehyde. Our third diagnostic uses potassium permanganate. Formic acid's structure gives it unique reducing power. When warmed, it reduces the permanganate, causing the purple color to completely disappear. Acetic acid lacks this reducing power, so the solution remains deep purple, providing a clear negative test. Reviewing the acidity diagnostic matrix, we can draw definitive lines of separation. Carboxylic acids react with weak bases like bicarbonate. Phenols are too weak for bicarbonate, but will react with a strong base like sodium hydroxide. Alcohols remain stable and won't react with either. Our specific laboratory results consolidate into this master table. The combination of effervescence, the unique buff and violet color complexes, and the reduction of potassium permanganate gives us a distinct cross-referenced fingerprint for salicylic, benzoic, formic, and acetic acids. Mastering organic chemistry laboratory work is not about memorizing a list of arbitrary reactions. It is about using rigorous logic and structural theory to unmask the unique identity of any compound on the bench.